Hello everyone, welcome to PW English YouTube channel. I'm Shalini Somashekar, your botany teacher. I welcome you to this lecture in the UMEED NEET English batch. Today we're going to study the last chapter in your 11th grade syllabus, which is plant growth and development in one shot. Before we get started off discussing the concepts, I'll quickly give you an overview of how many questions have been asked from this chapter in the last few years. As you can see here, according to the data, in the last few years at least, every year, 3 questions have been consistently asked from this chapter, which is just around how many questions you can expect in NEET 2025 as well. So, what about the syllabus? Should you follow the old NCERT or the new NCERT? Either of them will work. Some concepts from this chapter have been deleted after rationalization. It is in the old textbook. There are concepts like photoperiodism, vernalization, and seed dormancy. These are there in the old NCERT, but if you are preparing from old NCERT, ignore these concepts because these are deleted. Everything else is there. So as the name of the chapter itself indicates, plant growth and development, we are going to talk about how growth occurs in plants, what are the various features of growth in plants, and also we'll talk about how development takes place in plants. And then in the second part of the chapter, we will focus on plant growth regulators. Similar to how we all have hormones that control and coordinate our functioning and development, plants also produce chemicals that work like hormones in them. We call them plant growth regulators or phytohormones. We will learn about those groups of hormones and how exactly they influence growth and development in plants. Alright, so let's get started. First of all, what is growth? In the first part of this chapter, whatever you're learning, it's not something that you're learning for the first time. It's very easy to understand. Some bits and pieces of what you're learning here, you would have already covered in some other chapter already. So it's pretty chill, easy going, easy to understand, easy to remember and all of that. Okay. So what is growth? Where have we learned about growth? We've learned about growth as a characteristic feature of all living organisms in the chapter, The Living World, the first chapter, right? So growth is a characteristic of living organism. All living organisms grow and increase in mass or increase in number, right? That is what growth is. And it is a fundamental characteristic of life and also a very conspicuous, easily visible characteristic of life. When you look at culture of bacteria, if initially there were 100 cells and now they've become like 200, 250, 2000 cells, of course they have shown growth in terms of increase in number. If you look at us, like from when we were children to how we are now as adults and young adults, you know that we have grown in size, we have increased in mass. So that is also, very conspicuous, very easily visible. So growth is one of the most fundamental as well as very obvious characteristics of life. Like if we talk about metabolism, that is a characteristic of life. You can't really see it. It's not very conspicuous. It's something that's happening inside our cells at the cellular level. But growth is something that everybody can easily see with our naked eye, right? So that is what conspicuous word means. It's very evident. Okay, so that is growth. How does one define growth? We've also learned about this in the chapter, The Living World. Growth can be defined as an irreversible, that means you cannot reverse it, it is permanent, increase in the size of an organism or its part, one individual cell increasing or an organ increasing, increase in mass can be considered as growth, increase in the number of cells also is considered as growth. What does irreversible here mean? That means you can't go back to it. Like right now, if we are fat, you lose some fat and then you become lean. That we're not talking about that. We're talking about how growth happens from when we were little children to how we are now. You can't go back to becoming a baby today, right? A mango tree cannot go back to becoming a seed. It cannot become a small sapling. So that is irreversible and it's a permanent increase in the size of the organism. That is what growth is. All right. Now, how does growth happen? You know, in all living cells, metabolic activities keep taking place. Metabolic activities You have building up reactions, proteins are being built in the cell, carbohydrates are being built in the cell, like whatever food we eat, it gets broken down and it gets broken down into its monomer units, and later again they will get built up. Like in our cells constantly DNA replication is happening, which means DNA is making an extra copy, so DNA is getting synthesized, carbohydrates, probably glycogen is getting synthesized, and proteins are getting synthesized. So all of these are synthesis, building up reactions. These building up reactions are called anabolic reactions. Likewise, in every living organism, mostly since we're talking about plants, let's take the example of plants. If in plants you say photosynthesis is an anabolic reaction, plants make use of sunlight, carbon dioxide, we've learnt about it, and synthesize glucose, and then glucose is converted to sucrose or starch. So synthesis is happening. And catabolism means breakdown. Whatever food is synthesized, you know how it gets broken down during respiration to release energy, right? So anabolism is also happening, catabolism is also happening. Whenever anabolism is more than catabolism, whenever building up is greater than breaking down, then the organism tends to grow, right? So the organism grows. How can you measure growth when it comes to a plant? If a root has increased in length, that is growth. If the stem has grown taller, that is growth. If the stem has grown wider, thicker, that is also growth. If a leaf that was small has expanded and increased the surface area, that is also growth. So there are multiple ways in which you can measure growth in a plant. All right. Now, let's understand some characteristic features of how growth is with respect to plants. We know growth is common for all living organisms. Here we are studying about how is it unique to plants. What are the features of growth in plants specifically? In plants, we say that growth is indeterminate. We say that growth is unlimited, which is kind of unique to plants when you compare plants and animals. Of course, we also grow, but our growth is limited. You might say then we increase in size. I'm not talking about that. That is not irreversible growth, right? In a plant, why is there unlimited growth? Because they have special types of tissues called meristematic tissues, about which we've discussed in length, in detail in the chapter Anatomy of Flowering Plants. These cells always retain their capacity to divide, and they keep on dividing and give rise to more cells, right? So that kind of growth, unlimited growth, can be seen in plants because of the presence of meristematic tissue. Its only job is to keep dividing. Now, if you take a plant, for example, a tree, if a leaf falls off, a new leaf can form. If a branch gets cut off, or we ourselves cut it down, it will form newer branches. It has that ability. We don't have that ability. If our arm gets cut off, you can't develop a new arm, right? If your eye gets removed from your body, our body lacks the ability to synthesize that part again, which is not the case with plants. They show unlimited growth. Unlimited growth, when I say plants show unlimited growth, some of you might think that unlimited growth in terms of its length or height, like it's not growing tall permanently. It's not like one day the tree will reach the clouds and touch the clouds. That's not what I'm talking about. When I say it's unlimited, when I say it's indeterminate, I'm saying that if a leaf falls off, if all of the leaves in a tree fall off, the tree still retains the ability to grow them all back again. That is possible because of constantly dividing, rapidly dividing meristematic cells present in the plant, which we do not have, right? So over there it is unlimited, and the reason it is unlimited is because of the presence of meristems. Now, yeah, so all right. All the time, because of the presence of meristems, newer cells will keep getting added to the plant. At wherever regions in the plant meristems are present, newer cells are constantly going to keep getting added. Therefore, we call it open form of growth. Now, where do we find meristems in a plant? We've discussed this in the chapter Plant Growth and Development as well. We spoke about three different types of meristems based on their location. We spoke about apical meristem, which is present at the tip of the root and the tip of the shoot. Shoot apical and root apical meristem. We've spoken about intercalary meristem, which is present between the permanent tissues. We've also spoken about lateral meristem briefly. So it is present towards the side in dicot plants. So these are the different types of meristems. If you have to talk about the types of growth in plants, there are two different types of growth, primary growth and secondary growth. Primary growth basically is the increase in length of the plant. You can see primary growth in all plants, monocots, dicots. You can see it in both, in angiosperms. But secondary growth is increasing in girth of the stem or girth of the plant, like increasing in diameter. This is something that you can see only in dicots. You don't see them in monocots. Think about it. If you look at like a monocot plant like sugarcane or maize or rice or ragi, you never see the stem become trunks like a mango tree or a banyan tree, right? Even bamboo for that matter, it's a monocot. It does not increase in girth. As opposed to that, if you look at a dicot plant, it will undergo secondary growth generally. You can see secondary growth in dicots and it will grow wider, right? That is secondary growth. So, primary growth usually happens due to the apical meristems, where plants increase in length, which we call elongation of the plants along their axis. Lateral meristems, which is our cambium, vascular cambium and cork cambium, that is present only in dicots, that will contribute to an increase in girth of the stem, and as a result plants show secondary growth. All right, primary is because of apical mostly, and that's increase in length. You see that in both monocots and dicots. Specifically in some dicots, what you can see is increase in the girth of the stem and root as well, and that is due to the presence of lateral meristems. Example for that is vascular cambium and cork cambium. This is present only in the dicots. That is why you can see secondary growth only in the dicots. All right, now another thing, of course, growth is something that can be measured, right? Growth in plants also can be measured. In the beginning of this session itself, I told you what are the different ways in which you can measure growth in plants. It could be increase in the height of the plant, increase in the length of the root, increase in the surface area of the leaf. There are many parameters which you can use to measure growth in plant as well. Actually, why is growth happening? At the cellular level, what is happening? Growth can happen because of two reasons. One cell was there, it became two cells became four cells, then 100 cells gave rise to 1000 cells. There's increase in the number of cells that is contributing to growth. Another way growth can happen is there were 100 cells in a plant, all of these 100 cells increased in size and volume, that contributed to growth, right? If you think about it that way, if this is like a cell, okay, it has its own cytoplasm and organelles and everything. I'm talking about a plant cell specifically. If this increases in volume, if the cytoplasm inside increases, the volume of the cytoplasm increases, the cell tends to become larger, right? There is growth of the cell. That is the reason, one of the reasons why you see growth in the plant. Now, it's not very easy to measure increase in cytoplasm of a cell. Initially, the size of the cell was small. The cytoplasm was this much. Now it has accumulated more cytoplasm, volume has increased, and the plant cell has this much volume of cytoplasm right now. That's something that is difficult to measure. What can you measure instead? We'll see where that cell is. If the cell is part of a leaf, if a cell in the leaf has increased in volume, obviously the leaf also would have increased in surface area. If a cell is present in a stem and it has increased in volume, obviously the stem also would have increased in size, either increased in length or increased in girth, depending upon which kind of plant it is. So whichever part of the plant that cell is in, if it has shown some increase in volume, obviously the part of the plant, the part of the plant that cell is in, that part also will show an increase, right? So that is easier to measure. It's easier to measure how many centimeters or millimeters a root has grown instead of measuring how much extra volume of cytoplasm does one cell have right now. So instead of measuring the actual reason why there is growth, we are going to indirectly measure what that growth will contribute to. Okay. So at the cellular level, what happens is there will be an increase in the amount of protoplasm. Generally, it is measured by some quantity which is proportional to it. Alright, like I already mentioned. Some of the parameters that we can measure is fresh weight or dry weight of any plant organ, root, stem, leaf. Length. If a pollen tube has fallen on the stigma, how long has the pollen tube grown. It was initially 1 millimeter, now it is 5 millimeters. So that is one way you can measure it. Area, in a dorsiventral leaf, what is the surface area that has increased? Volume, the cells in a watermelon fruit increases in size up to 3,50,000 times apparently. That means each cell accumulates more water and more cytoplasm and increases in size almost 3,50,000 times. So that can be measured. And also the number of cells. One single cell, meristematic cell, present in the root apical meristem of maize, in 1 hour alone, just in 1 hour, one cell will divide so rapidly and give rise to as many as 70,500 brand new cells. So, obviously in the root apical meristem, meristematic cells are constantly dividing, but this number is kind of mind-blowing. So, these are the different ways in which you can measure growth, either as the length or as the surface area, the cell volume, the number of cells, any of these parameters. Okay. Yeah. Now let's talk about phases of growth. Little bit about it you've already learnt in the chapter Morphology of Flowering Plants. Remember when we spoke about the different regions of the root, I mentioned there's a root cap and there's a meristematic area, there's a region of elongation, region of maturation. We've learnt about all that. So a little bit related to that is what we're going to learn here. In growth, in plants, you can typically see 3 phases. The first phase here is called the meristematic phase. Over here, the cells that are in the meristematic phase obviously will be present at the meristematic region, root tip, shoot tip. If it's dicot, then the cambium. So what happens is these cells are very small. Remember, their job is to only keep dividing. They don't have time to make a thick cell wall. They don't have the time to develop one large central vacuole. Their only job is to keep on dividing. They don't even increase in size that much. These cells are small and they have rich protoplasm. They will have a large conspicuous nucleus, okay? And the cell wall is primary, made up of cellulose, mostly thin and cellulosic. It will have a lot of plasmodesmata, that is the cytoplasmic connections between the cells. All right. Next, elongation. Remember, all cells in the plant don't have the ability to divide. At one point, some of the meristematic cells will lose their ability to divide and become permanent, like parenchyma, collenchyma, sclerenchyma, xylem, phloem. So what happens is, at one point they stop dividing, they lose their ability to divide. Let's say there were 100 meristematic cells, 50 cells lose the ability to divide. The remaining 50 will continue to divide, give rise to 100 again. Again 50 will, lose the ability to divide, and again they will take up some other permanent function in the plant. The remaining meristematic cells continue to divide. This is how it happens. See, the meristematic cells, they don't perform any other job other than dividing. But in a plant, there are so many functions to perform. Somebody has to transport water across different parts of the plant. Some tissue has to transport food across different parts of the plant. Some tissue has to perform photosynthesis. Some tissue has to provide mechanical support. There's a lot of work to be performed inside the plant. If all cells are just dividing, doing nothing, then it's not a good thing for the plant, right? So some of these cells stop dividing and take up permanent functions. One cell will be like, I want to provide mechanical support, and it will become sclerenchyma. Another cell will be like, I think I really might enjoy performing photosynthesis. It becomes like chlorenchyma in the leaf. Another cell might be like, I think I do a good job conducting water, and it probably becomes one of the cells in the xylem. So different cells stop dividing and take up permanent functions in the plant, right? So when they stop dividing, they enter a phase called the elongation phase. At this time, remember I told you when they were meristematic, they weren't growing in size that much. They were growing a little bit in size, divide all the time. Now that they're not dividing anymore, they have some time to grow. And they will grow in the longitudinal axis, so the cells will start appearing to elongate. As the cells appear to elongate, vacuolation also will happen. Formation of vacuole also will happen. Okay, so you can see vacuolation here. Cell will start to enlarge, and now that the cell is becoming bigger and bigger, you will need newer cell wall to get deposited. So new cell wall deposition also will happen. Then the third phase, the last phase, is your maturation phase. Here the cell would have decided what it will become, what it wants to become. what it wants to mature into. Does it want to perform photosynthesis? Does it want to transport water? Whatever it wants to do, it would have decided. And according to what that job demands, it will change itself. Like right now, if you want to be a good student, there should be some characteristics that you should show. For example, staying focused during a lecture, or taking notes, or being passionate about your goal. These are some traits that an ideal student should have. If I want to be like a good teacher, then I should have a good hold on the subject. I should understand the subject well. So there are some traits that a good teacher should have. So if one of these cells decide to become xylem, they should have some traits to become xylem. In xylem, if it has to conduct water, it should become a tracheal element like vessel, right? If it wants to become a vessel, it has to lose its cytoplasm. It should die. So according to what function it will perform, it will develop its skills. It will prepare and get modified accordingly. If one cell decides to, perform photosynthesis, then it should produce a lot of, it should have a lot of chloroplast within it where photosynthesis will take place. If it decides to be a cell which stores food, it should become parenchyma. So depending upon what job it is going to take up in the future, it will modify itself accordingly. Alright, so that process where the cell will modify in order to fit in that job that it's going to perform, whatever it does in that process, that is called maturation or differentiation. Alright, so over here the cell was growing. By the time it becomes mature, it would have reached its final size. It's no longer going to divide, or it's not even going to increase in size. Okay, so they will attain maximal size in terms of the cell and as well as the cell wall. Depending upon what it is going to form, it will modify the cell wall accordingly. If it has to become sclerenchyma, it should deposit lignin in the secondary wall. If it decides to become endodermis in the root, there should be suberin deposition in the wall. So depending upon what it is going to become, it will modify accordingly. Now there's this one diagram that is given to you in NCERT. What you're looking at here is a portion of the root. In the root, the meristematic zone will be like at the tip, very close to the tip. Following that, you will have elongation zone. Following that, you will have maturation zone. So if you have to identify where exactly the elongation zone is present, this kind of parallel line diagram will help you. So if you notice here, the same lines that are almost equidistant here are not equidistant anymore here. Okay, so if you see here, it is the distance is less. Here the distance is more, and if you look at this B, C, D areas, the distance is very more high. Okay, it's very large compared to the other regions. This indicates that this is the zone of elongation, where the cells have increased in size. Okay, so this is one way of finding out. Now, let's talk about growth rates. If you measure growth with respect to time, you will get growth rate. Whenever you're talking about rate, time will 100% come there. You're always measuring growth in terms of, if you're measuring growth in terms of how quickly it is growing with respect to time, then you call it growth rate. Okay, so growth rate is increase in growth or growth happening per unit time. This could be 1 day, this could be 1 week, this could be 1 month, this could be 1 year. It depends, depending upon how you are calculating it, what is your research requirement, you can change it. Okay, so the increased growth per unit time is what we call growth rate. Since it is with respect to time, of course, it can be expressed mathematically. Now, there are two types of growth rates. Growth rate can be measured in two ways. One is arithmetic growth rate, the other one is geometric growth rate. What happens in arithmetic growth rate, I'll tell you. Now, if there is one cell, let's say one meristematic cell is there, this cell decides to divide. Of course, it will divide because it is a meristematic cell. Its only job is to keep on dividing. One cell divided by mitosis gave rise to two cells. Now, of these two cells, one cell is going to get differentiated and lose its ability to divide. The other cell continues to divide. Of the two cells, one cell will again mature and lose the ability to divide. This will continue to divide again. So growth is happening linearly. At each mitotic division, exactly 1 cell is getting added to the plant. Right? Look here. This is how it is given in NCERT. You have one cell that divided into two. Of these two cells, one cell become differentiated, that is represented by light blue. This continued to divide and gave rise to two. Again, of these two, one differentiated. See, there is increase in size and differentiation, elongation, differentiation has happened. So at each step, you can see how one cell is getting added. Here you had 2, here you have 3, here you have 4, next you have 5, next you have 6. So in a linear way, growth will take place over here. And if you represent this on a graph where you have time on the x-axis and, for example, height of the plant on the y-axis, it could be the length of the root, length of the stem, height of the stem, whatever. Length is usually arithmetic. That is how it grows. So you can see how in the graph you will get a straight line. At a constant rate, it is getting increased. Okay, if you have to express this mathematically, this is the equation where LT represents the final length, L0 is the initial length when we started calculating, R is the rate of growth, which is arithmetic growth rate, and T is the time. If the growth rate with respect to time, if per second is growing, increasing 2 centimeters or 2 millimeters, then you will get one final length. If per second, if it's increasing 4 millimeters, then you'll get a different final length. It will be much higher. So how quickly growth is happening, depending upon that, the final length will change. Okay, so when you started off, let's say it was just 1 millimeter, the root. With every passing second, per second, in one case, 2 millimeters is increasing, in the other case, per second, 4 millimeters is increasing. The time duration is the same, but the increase in the length is greater here, less here. So this will definitely have a higher growth rate. This will have relatively lower growth rate. If the growth rate is high, then obviously the final increase in the length also will be more. Ultimately, this will be the same, initial length will be the same. How much of an increase you see in final length depends on this growth rate. If the growth rate is higher, then you will see higher increase in the length of whichever plant part you're considering. It could be the root, it could be the stem, whichever one you're considering. Okay, so that is arithmetic growth. Then we have geometric growth. In geometric growth, what happens is, you have one cell that gives rise to two cells. Both of these cells retain the ability to divide. They further give rise to two cells, which will again undergo cell division. Again, these two cells retain the ability to divide. They continue to divide, and this goes on. So exponentially, you can see there is an increase in the number of cells. So initially, how many were there? There was only 1, 2 to the power 0. Then you have 2 to the power 1, 2 are there. Then you have 2 to the power 2, 4 are there. Then you have 2 to the power 3, 8 are there. So exponentially, you can see an increase in the growth. That is called arithmetic growth. Now when you plot a graph, that is called geometric growth. Now when you plot a graph for geometric growth, having the same thing, time on the x-axis, and over here if you have, take for example, the size or the weight of a plant organ on the y-axis, what we get here is an S-shaped curve. We call this the sigmoid curve. Because it is S-shaped, we call it the sigmoid curve. Have I mentioned it anywhere? No. Okay. So this is the sigmoid curve. In this sigmoid curve, you can see three very distinct faces. You have a lag face here. You have a log face here. Okay. And then You have a steady phase or a stationary phase here. What can we see in the lag phase? Is there growth at all? There is growth. Initially it was this much, the size or the weight, but right now it is this much. Growth has been there, but it is a little slow. Slow growth happens in lag phase. Doesn't mean growth is not happening at all, it's happening but a very low rate. What happens in exponential phase? Exponential phase, here itself the geometric growth will take place. Over here at an exponential rate, growth will happen. This is also called as the log phase. I want you to notice how if you take from here to here as one unit, take a similar unit from here to here next. When you started off here, growth was here. Now check at this point, it is here. In the same amount of time, in the lag phase, you saw only this much growth in this much time. In the same amount of time, I want you to see how much more, almost thrice or 4 times more growth you can see in exponential phase. So that is where geometric growth is taking place. Geometric growth rate applies to this portion of the sigmoid curve. And then finally, there is stationary phase. A plant cannot, afford to continuously keep growing in the exponential phase forever. Probably one or the other thing will become limiting. The resources might become limiting, or some inhibitory hormones might start acting upon that plant part. So its growth will become stagnant. Okay, so that is called the steady phase. It will not increase anymore. So first, in the lag phase, the growth is slow. Growth is there, but it is slow. Exponential phase is called the log phase as well. That is the second phase. Here, growth increases at an exponential rate. Both the progenies, like I told you, both the progeny cells will continue to divide and keep adding on more cells. Then finally, we have the stationary phase, which is also called the steady phase. Here, growth will slow down because nutrients probably become limiting. Okay. Now, if we have to represent this in an equation, okay, it's mentioned here, sigmoid or S-shaped curve. The equation that we use to represent this portion of the graph where geometric growth is seen, this is the equation. W1 is equal to W0 e to the power RT, where W1 is the final size or weight or whichever parameter you're using. W0 is the initial size. R again is rate, geometric growth rate. T is time. E, because it's happening at an exponential rate, we bring in E as well. E is nothing but the base of natural logarithms. Okay. Now, here is an illustration from NCERT. When the zygote is first formed, everybody knows the zygote, right? In all sexually reproducing organisms, the first cell of our life is the zygote. When the male gamete fuses with the female gamete, gives rise to a diploid cell, that is the zygote. Even in plants, the zygote is formed, right? And sexually if they're forming by sexual reproduction, then zygote will form. The division of the zygote initially will be in the exponential phase. Eventually, some of the cells will divide exponentially. Some of the cells will show arithmetic growth. So initially you can see how 2 cells were there, both of them divided. 4 cells were there, all of them divided to give rise to 8 cells. But as it moves on, some of the cells retain the ability to undergo geometric growth, like the ones in dark blue, and the some of the cells don't. Okay, so eventually some of the cells will start dividing according to the arithmetic growth, and some of the cells completely lose the ability to divide and become permanent tissues. Alright, so that's what happens. Now let's talk about the different ways in which you can compare growth. There are 2 different types. One is absolute growth rate, the other one is relative growth rate. Okay. So if you want to compare the growth rate in 2 different plants or 2 different parts of the same plant or anything, if you want to compare growth in plants, then one is absolute growth rate, the other one is relative growth rate. Okay. So first let's understand absolute growth rate. Here we have 2 leaves. This leaf is called A and this leaf is called B. This small leaf's initial surface area is 5 centimeter square. This leaf's surface area is 50 centimeter square. Okay, after the same given period of time, say X period of time, they will measure the growth in both these leaves again. Initially it was 5 centimeter square, now it has increased in surface area and become 10 centimeter square. This also has increased in surface area. What was initially 50 is right now 55 centimeter square. Let's call this B1 and this A1. Now if you calculate how much of an increase has happened over this period of time, you can see that in leaf A there has been a plus 5 centimeter square increase in area, and over here also there is plus 5 centimeter square increase. So if you think about it, the absolute growth rate is the same in both A and B. Both of them have shown an increase in 5 centimeter square. If you compare that with the initial size, initially it was 5, right now it is 10. So over here you can see that the surface area has doubled. It has increased, right? Let me write it as into 2 instead of 2x. Okay, so this has doubled here. But over here, if only it increased at the same rate, you should have seen 50 centimeter square becoming 100 centimeter square, which is not the case. It has increased by how much? By 1/10, right? So here you can see 1/10 is 0.1% only increase. Here you can see 100% increase. Here 100% increase, it has doubled. Here only 0.1% increase. So in that way, if you take the relative densities, the relative growth rate, A has shown a greater relative growth compared to B. Okay, so absolute growth rate is a measurement and the comparison of the total growth that has happened per unit time. Relative growth rate, the growth of the given system, which could be any part of the plant per unit time, expressed on a common basis. For example, how much was it initially? If you compare it, then you get relative growth rate. Relative to how much was present initially, how much increase is it showing? Has it doubled? Has it tripled? Has it increased 1/4? So that will give you the relative growth rate. Okay, that's one way of measuring it or comparing the growth in two different entities. Now, what are some conditions that are required for growth? Of course, water is very, very important. When I told you a cell that is smaller increases in size, accumulates more protoplasm. What I mean is, for that kind of enlargement to take place in a cell, water is essential. When water moves into the cell, let's consider this as the plant cell. When water moves into the cell, the protoplasmic or the cytoplasmic contents will increase, and as a result of increase in the volume of the cytoplasm, the protoplasm will exert a pressure on the cell wall. Remember, plant cells have cell walls. So that is called turgidity or turgor pressure. Okay, so whenever water moves into cells, the cells will become what we call as turgid. When the cell becomes turgid, it will exert a pressure on the cell wall, and that is what will help the cell to grow in size. Okay, so this turgidity is essential for growth of the cells. Okay, and since cytoplasm is mostly water, it also acts like a medium where many metabolic reactions take place. All right, next, oxygen. Why is oxygen important? We've learned about it in the previous chapter itself. For generating ATP, we need oxygen. If a cell has to grow, it requires energy, and that energy comes from ATP. So oxygen is necessary for breaking down food molecules and producing energy that a plant requires to grow. So oxygen helps in releasing metabolic energy that is essential for growth activities. Next, for plant growth, many different nutrients are required. What are nutrients for plants? The minerals that it absorbs from the soil, such as nitrogen, potassium, phosphorus, boron, silicon. and magnesium, iron, all of these are essential for the growth of plants. Okay, so some of them are required in slightly larger quantities, we call them macronutrients. Some of them are required only in traces, we call them micro nutrients. So it's like, if not for these macro and micronutrients, plants cannot grow to their full potential. Okay, so macro and micro essential elements are required by plants for the synthesis of protoplasm, and they also act as a source of energy. Next, temperature. Every plant will have its own temperature optimum, all right, depending upon where it generally grows. If it grows in tropics or temperate regions, depending upon that, it will have its own temperature optimum. That is important because the enzymes of that plant functions very well in that temperature. If there is any major deviation from this temperature, the enzymes, if it's too hot, the enzymes could get denatured. If it's too cold, the enzymes could become inactivated. So if the temperature is not optimum, the enzymes cannot function properly. As a result, growth also may not happen to the best extent. Then environmental signals such as light, gravity also affects growth and development. You know, some plants need a specific duration of light, specific duration of darkness to flower. Okay, so roots need to know about gravity in order to move towards the soil, grow towards the soil. Some plants flower only after a cold treatment. So for their development also, it is essential, these environmental factors. Remember at the beginning of the session, I told you three concepts have been deleted: photoperiodism, vernalization, and dormancy. Actually, photoperiodism talks about how certain plants depend on specific length, day length, specific duration of darkness in order to flower. Vernalization is about how plants, require a cold treatment prior to flowering. So it depends on all of those environmental factors as well. So that's about conditions required for growth. Now let's talk about an important concept: differentiation, de-differentiation, re-differentiation. You might already know what is differentiation. I've told you about this in the chapter Anatomy of Flowering Plants. I've told you about this today as well. So you have meristematic cells. Okay, let's write meristematic tissue. This meristematic tissue stops dividing. Once it stops undergoing cell division, it loses the ability to divide and take up certain permanent function in the plant. Then we say that it has become mature, right? It becomes a permanent tissue. Now, the process by which the meristematic tissue became a permanent tissue is called differentiation. Okay, now most times, majority of the times, once a tissue becomes permanent, it will for the rest of its life continue to do that job. If it has decided to become chlorine-chyme and perform photosynthesis, for the most part it will continue to do that. If it has decided to become like a regular parenchyma and store food, then it continues to do that. If it has decided to become anything else, it will continue to do that. But sometimes during some conditions, during certain conditions, especially in dicots, what we can see is these plants that have undergone differentiation, these tissues that have undergone differentiation completely stop dividing, regain their ability to divide. Okay, let's say for example, this permanent tissue, it had become parenchyma. After differentiation, let's say it had become parenchyma. Okay, now if a permanent tissue that has lost the ability to divide regains the ability to divide and become meristematic again, this process is called de-differentiation. Now what we get here is a meristem that we call differentiated meristem. This wasn't always a meristem, it had become permanent, but right now it has regained the ability to divide and become meristematic again. So, because it's happening by a process called de-differentiation, we're going to call this de-differentiated meristem. Now, this de-differentiated meristem can become something else again. It now is open to either become parenchyma, collenchyma, sclerenchyma, whatever it wants. Okay? Just because it was parenchyma earlier doesn't mean it has to become parenchyma again. This meristematic tissue can undergo differentiation again to give rise to another type of tissue, entirely different type of tissue as well. So this can again become a permanent tissue. Like let's say xylem. It could become xylem, it could become parenchyma also again. It doesn't necessarily mean that it has to become something new. It could become something new or it might differentiate to become parenchyma again. Anything can happen. Okay. So this de-differentiated meristem becoming permanent again by losing ability to divide. This process is called redifferentiation. This process is called redifferentiation. Differentiation is first time becoming permanent. De-differentiation is regaining the ability to divide and becoming meristematic again. After that, one more time undergoing differentiation. One more time means repeated, right? One more time if it's getting differentiated, then we call it redifferentiation. All right. Now, let's talk about development. When a plant grows, we know that there is increase in the number of cells. There could be an increase in the size of cells. When a plant develops, what happens? We talk about specific to angiosperms. From the time the seed germinates, whatever changes take place, the seed germinates, becomes a small sapling, becomes a small plant, then probably becomes a tree, and then it ages. All of these processes, from the time it germinates to the time of its aging and death, all of the processes that take place together, we call it as development. So what are some of the processes that take place during development? Initially, you have meristematic cells. Meristematic cells increase in their size, plasma growth happens, and they undergo cell division. Some of these cells retain their ability to divide and stay meristematic. Some of these cells will undergo differentiation. They will also show elongation. First, there is increase in growth of cytoplasm, plasmatic growth. As a result, they will elongate. Eventually, they will mature, get differentiated and become mature. Ultimately, they will become the mature cell. And this mature cell will undergo aging. Aging is called senescence. And ultimately, death will happen. So, during the development, all of these processes is going to take place. All right. Now, let's talk about a concept called plasticity. What is plasticity? Plasticity basically is, with respect to plant growth and development specifically, when a plant is developing, it will have more than one option. Okay. Now, it could develop according to pathway 1 or it could develop according to pathway 2. Now, which pathway this plant takes to develop could depend on two things. Either it could depend on which phase in its life that plant is. Is it very young, juvenile, or is it mature? Or it depends upon the surrounding environment. For example, there's one plant, it has two developmental pathways. If it is present in terrestrial habitat, it will take one pathway. If it is present in aquatic habitat, the same plant, the same species will choose another pathway. Likewise, there is one plant. It has one developmental pathway for its juvenile stage, another developmental pathway for its adult phase. If it is that juvenile phase, it will take one developmental pathway. If it is adult, it will take another developmental pathway. So plants have the ability to follow different pathways. We think plants lack brain and they're stupid, but plants are super intelligent. They follow different pathways in response to either environment or its phase of life to form different kinds of structures. First example, how you can see differences in different phases of life. Here you can see two types of leaves will be formed. That's why it's called heterophyly. Phyll means leaf. Hetero means many. More than one type of leaf. If the plant is forming, then we call it heterophyly. You can see this in plants like cotton, coriander and larkspur. So over here, the juvenile plant leaf is very different from the mature plant leaf. Next time you buy coriander for your house from the market, I want you to notice how the shape of the leaves is different in the younger leaves and in the older leaves. In response to environmental factors, there's one plant called buttercup that can grow in terrestrial habitats and also in aquatic habitats. When it grows in terrestrial habitats, its leaves are like this. When it grows in water habitats, its leaves are more dissected, like this. The reason why it's more dissected is because if it's growing inside water, because of the water currents, the leaves should not tear up, right, and get damaged. So they themselves have leaves in such a way that it is not vulnerable to tearing. Okay, so that is the second example for plasticity. That's about the first part of the chapter, growth and development. Now it is time to talk about plant growth regulators. Plant growth regulators have other names as well. They are also called plant growth substances, plant hormones and also phytohormones. These plant growth regulators are very diverse chemically. It's not like every plant growth regulator is a protein. It's not like every plant growth regulator is an indole compound. What do we call a plant growth regulator? Any chemical in the plant, any chemical that has an ability to regulate the growth and development in a plant. If there is any chemical that can increase the growth rate in a plant or retard the growth rate in a plant, that chemical is a plant growth regulator. What is the regulator? You've seen regulator in your gas stoves. You've seen regulator right next to the fan switch where you can control the speed. You can either increase or you can decrease. You can increase the flame in your gas stove or decrease it. Likewise, regulators are not always going to promote growth. Some of them could promote growth and development and cause the plant to grow, leaves to grow, chloroplast to develop, while some others will cause a retardation in the growth. They will not stimulate growth, they will stimulate dormancy. Okay, so whichever chemical has the ability to either promote or retard the growth of a plant, such chemicals are called plant growth regulators. Alright, they are very diverse because the only function for us to call a chemical as a plant growth regulator is that it should show some influence on the growth and development of the plant. You have many such chemicals, okay, so they're obviously very diverse. We have one group of plant hormones called auxins. These are derived from indole compounds. One example for that is indole acetic acid, which we abbreviate as IAA. Then we have another group of hormones called cytokinins. These are derived from adenine. You know adenine, right? In biomolecules, you've learned adenine, thymine, guanine, cytosine. It's a nitrogenous base. So it is derived from that. Then gibberellic acid, we also call it gibberellins. They are terpene derivatives. Ethylene is a gaseous plant growth regulator. Abscisic acid is derived from carotenoids. You know what carotenoids are, right? They are pigments. So these are the different groups of chemicals. So they are very diverse. Now let's talk about its classification. Plant growth regulators can be classified into two categories. The first one being growth promoters and the second one is growth inhibitors. Very clearly, if the chemical, the plant hormone shows growth promoting activities, then you call it a plant growth promoter. For example, cell division. If more cells are formed because of cell division, obviously there will be growth. Cell enlargement, cell increasing will contribute to growth. Pattern formation, tropic growth, formation of flowers, formation of fruit, seed formation, all of these are activities that are promoting growth and development. So if a hormone has an influence on any of these functions, then we call it a growth promoting hormone or a growth promoter. On the other hand, we have growth inhibitors which inhibit growth in plants. For example, they could cause dormancy. Dormancy means not allowing the seed to germinate, making the seed become dormant, or abscission, where some parts of the plant fall off, right? Like the older leaves fall off and the fruits fall off. So you might be wondering, why do plants produce chemicals that inhibit the growth of the plant? Why should a plant produce a chemical that will inhibit seed germination? If the seed germination doesn't happen, then the next generation of plants will not be produced. Why would a plant not want its next generation to be formed, right? I'll tell you when these plant growth inhibitors function, okay? So, you know how your parents are very protective of you all, right? Even I'm very protective of my child. I do not want my child to go to an area which is not safe for him, right? So if there is a very heavy traffic, I will not allow my son to play there. If if the area is not safe for my child, I will not allow him to play there. Likewise, every organism on our planet, regardless of whether it has a brain or not, including plants, are very, very cautious. They want the best for their children. What is the baby for a plant? It is its seed, right? It wants the seed to grow, develop well, survive, and then produce its own flowers and fruits and be healthy, right? If the condition surrounding this plant is not favorable, if there is not sufficient water in the soil, if the temperature is too high, then the parent plant will not want the seed to germinate at such a time. If a seed germinates at a time when the conditions are not very favorable for it, it's not likely to survive. In that very high temperature, that small sapling could get dehydrated and die. If there is not sufficient water, the plant could die, because younger plants, similar to how in humans babies are more vulnerable to get infections compared to adults, even in plants, younger plants are more vulnerable to these kind of extreme external climatic conditions. So under such cases, plant growth inhibitors will make the seed dormant. When the favorable conditions are there, activity will be promoted and the seed will germinate. Abscission. The plant will not make young green leaves to fall off. Older leaves will be made to fall off because older leaves cannot function optimally anymore. What is the point of giving food to a leaf if it is not performing photosynthesis for you, right? It has to perform its job. If it is not able to perform its job well, it will have to be let go of, right? So that will fall off. And what is the point of holding on to a fruit forever? What is the function of a fruit? Function of a fruit is to help in seed dispersal, right? Seeds have to be dispersed. Only if a fruit falls off a tree, then only newer plants can arise from the seeds. If the fruit is forever attached to the plant, then it serves no purpose, right? Whenever it is the right time for this leaf to fall, for the fruit to fall, the plant has to make sure that those parts fall off, right, for its own good. That is when these growth inhibitors function. They are the good guys only. Okay, just like your parent has a side where they pamper you a lot and there's another side where they're very protective of you. Similarly, there are plant regulators that promote growth and there are plant regulators that kind of inhibit growth. Both of them are essential. Just because they are inhibiting something doesn't mean they are bad for the plant. They are inhibiting something because they know that it is not good for the future of the plant. Alright. So among the hormones that we studied about now, auxin, cytokinins, gibberellins, ethylene, abscisic acid, the first three are promoters, plant growth promoters. Abscisic acid is a plant growth inhibitor. Ethylene has both promoting as well as inhibiting activities, but a little bit more of inhibiting activity. Okay, now we have to learn about its discovery, how all of these plant growth regulators were discovered, and also about how do they function inside the plant. Now what's interesting here is all of these plant growth regulators were discovered accidentally. There was no scientist or researcher trying to find out which chemical has growth regulating properties in plants, or can this chemical be a plant growth regulator. Nobody was worried about that. They were not discovering that. They were not working towards it. They were working on something else, and accidentally they found out, okay, this chemical has this effect on plants. Okay, so we'll have to learn about the discovery story of all of these plant growth regulators, and also how exactly they function, how they regulate the growth and all of that. First, let's start off with auxins. Okay, so auxins were discovered by Charles Darwin and his son Francis Darwin. Everybody knows Charles Darwin for his work in the theory of evolution, right? Origin of species, we know about that. But Charles Darwin and Francis Darwin, they were also interested about how plants function, the physiology of plants and all of that. They had noticed that plants bend towards sunlight. You've all done this experiment in your school where you have a plant and then there is a cardboard box outside it with one hole cut out on one of the walls of the cardboard box, and then the plant tends to bend and come out of that wall, come out of that hole, right? It bends towards sunlight. They were trying to understand what exactly happens there. So then what they did was they were working on this one kind of monocot plant called canary grass. And they studied the coleoptiles of canary grass. What are coleoptiles? You know from morphology of flowering plants. I've told you that within the seed, okay, let me draw like a monocot seed. There will be endosperm and there will be embryo here, and in the embryo you will have a plumule, and on top of that plumule there will be a sheath called the coleoptile, right? We're talking about the same coleoptile. They were studying the coleoptile. They noticed that whenever there was sunlight only from one direction, unilateral means only from one side, if there was illumination of light, they would always grow towards the source of light itself. We call that phototropism, positive phototropism, because it moves towards the light. They noticed that this coleoptile also would move towards light. Then they concluded by saying that there is some chemical that is present at the tip of this coleoptile that is causing it to bend towards sunlight. What they did to confirm their findings is they took another coleoptile and cut off its tip. We call that decapitation. Whenever they performed decapitation and removed the tip, they did not show any bending. Again, when they removed the tip, placed a sheet of gelatin, and on top of that, when they placed the tip again, through gelatin, the chemical could pass, and then it showed bending again. So they concluded that there is some chemical that is present at the tip of the coleoptile which is causing it to bend towards light. But they did not isolate what is that chemical. They did not find out about its chemical nature. That was first isolated by a person called F.W. Wendt. And he did not isolate that from canary grass. He isolated that from oat seedlings. You know oats, right? That diet food that everybody eats. Yeah, from those seedlings. Now, what are auxins? Where is this word derived from? Auxin in Greek means to grow. It was isolated from human urine. How did it get to human urine? Because we eat plants, and whatever chemicals are there in plants will get into our bodies and they can get excreted in the urine. So it was first isolated from human urine. Where is it generally produced? It's produced at the tip of the plants, generally the growing regions of the plants, like the root tip and the shoot tips. Okay, and what are the chemicals that we call as auxin? There is one auxin that is naturally produced inside plants, which is an indole derivative, which is called indole acetic acid. Remember, I gave you this example for auxin initially. This indole acetic acid, whatever effects indole acetic acid has on a plant, if there is any other chemical in the world that shows the same kind of effect on a plant, you can call it an auxin. Not all of these auxins are produced by plants. Some of them are naturally synthesized by plants. Some of them are produced chemically also. So, indole acetic acid is natural. Similarly, you have another auxin which is natural, which is indole butyric acid. IAA, IBA, these are natural. But there are some synthetic auxins as well, like naphthalene acetic acid, which is not produced by the plant itself. It is chemically synthesized in the labs. And you have something called 2,4-D, which also shows activity similar to IAA. 2,4-D is for 2,4-dichlorophenoxyacetic acid. Okay. Yeah. Now, let's learn about the physiological effects of auxins. Auxins are used to initiate rooting in stem cuttings. You may have noticed how in plants like rose and jasmine, stem is cut and then you come and plant it in your house. It becomes an entire rose plant. Vegetative propagation can be done by just using the cuttings of stem, right? In such cases, if you allow the stem to be inside the soil, naturally itself after some time roots will start forming. But if you have to make that process happen rapidly, then by applying a paste of auxin at the base of that cut stem, if you keep it then inside the root, inside the soil, then the rooting will happen much quicker. So it helps in rooting of producing roots in stem cutting. Secondly, what it promotes is apical dominance. You see, in a plant, you have apical bud, or it's also called terminal bud, and you have these buds at the axil of the leaves, which we call axillary or lateral buds. Now, for as long as apical bud is there, that is where the auxins are located, remember. Because of the presence of auxins, these buds will not be allowed to grow, they will not be allowed to branch. Only lateral growth or increase in length will be taking place. The plant will not become bushy. What will happen when you cut out this part is, then once the apical bud is removed, then the lateral buds will start growing and branching out. Okay, so for as long as auxins are there, it promotes apical dominance. Despite the presence of lateral buds, this will dominate and increase in height, increase in length, that is called apical dominance. Second, it prevents early leaf and fruit drop. It will prevent young leaves which are functioning well to drop. If a fruit is not fully developed, the seeds are not developed, what is the point of it falling down? If it falls down, the seed cannot germinate and become a new plant. So when it is not time for the fruit to fall, when it is not time for the leaf to fall, it will prevent it from falling. But the same auxins will promote the abscission or falling off of old leaves and mature fruits. Okay? But it promotes abscission of older mature leaves and fruits. Specifically in pineapple plants, auxins promote flowering. Specifically in tomatoes, it induces parthenocarpy. Parthenocarpy means forming seedless fruits. It promotes cell division as well. It is used in tissue culture along with cytokinins. Okay, it controls the differentiation of xylem, very important. It acts as a weedicide as well. Specifically 2,4-D, a synthetic auxin, is used as a weedicide. It kills only dicot plants. So if you have a field full of monocots, you're either growing ragi, jowar, or rice or sugarcane, all of these are monocot plants, and you have some dicot weeds here and there, and if you want to get rid of them, if you spray 2,4-D, it will not affect the monocots, it will specifically target and kill the dicot plants. Alright, now, talk about gibberellins. The story of gibberellin discovery is kind of nice, interesting. Gibberellins were discovered by Japanese researchers. You know, in Japan, also similar to how in India we grow a lot of rice, especially in South India we consume a lot of rice. Even in Japan they consume a lot of rice. They grow rice a lot. And apparently there was one disease that they could find in rice saplings, which they called the Bakane disease. Bakane in Japanese means foolish. Okay, so Bakane disease loosely translates to foolish seedling disease. Now, why are they calling these rice saplings or seedlings as foolish? Why are they foolish? Because they are growing unusually tall, taller than normal. And their leaves are also kind of yellow. They are not capable of performing photosynthesis efficiently. And also, they themselves cannot bear their own weight and they are toppling and falling off. So, these are the characteristics of the Bakane disease. It was acting like a fool because it was just growing so tall it cannot bear its own weight and it would fall off. Okay, so they were studying what is causing this Bakane disease in rice. Okay, now apparently this disease was caused by a fungus called Gibberella fujikuroi. This one person, Japanese researcher called Kurosawa, he reported that whenever healthy plants, healthy rice seedlings were treated with sterile filtrates of this fungus. Let's say there is one rice plant that is healthy and there is this rice plant that is growing unusually tall. It's not producing proper kind of leaves. Inside this, you have Gibberella fujikuroi, which is causing the disease. If they removed this yellow color Gibberella fujikuroi and injected that to a healthy plant, this also would show the symptoms of disease eventually. Okay? He reported that the appearance of symptoms of the disease in uninfected rice seedlings when they were treated with sterile filtrates of this fungus. Later, they identified in this fungus, which is that chemical in the fungus that is causing this effect, they identified it to be acidic in nature and they called it gibberellic acid because it was obtained from Gibberella fujikuroi. They called it gibberellic acid because it was acidic in nature. Alright. Now, this is a growth promoting PGR. I've already told you about that. So far, more than 100 PGRs have been isolated, but the most popular one and the most used one is GA3. You don't give it fancy names. You just call it gibberellic acid 1, gibberellic acid 2, gibberellic acid 3. GA3 is the most popular and the most widely used and the most studied also. It's obvious that they're called gibberellic acid because they are acidic in nature, right? Now, what are its physiological effects? You know that in the rice stem, in the rice plant, it increases the stem size. For a rice, it may not be beneficial. But for a plant like sugarcane, where the commercially important part itself is the stem, this chemical can be used to increase the stem so that the product that you get from that plant, the harvest will be higher. Okay, so mostly the effects of gibberellin is with respect to increase in length of one or the other part. In sugarcane, it is used in the increase of length. In plants with rosette habits like beet and cabbage, it promotes bolting. Bolting means increase in the internodal length just before flowering. So that is one effect. In juvenile conifers which have not yet produced seeds, it will fasten the maturity period and give rise to the seeds. With the seeds, they can get next generation, and in some conifers, seeds are consumed as food. It's considered as a delicacy. So they will get their product earlier. Next, in grape fruit, it increases the length of the stalk so that there is less crowding and grapes will get sufficient amount of space to grow and they will not get spoiled soon. Okay. Next, in apple, it will elongate the shape to make it aesthetically good looking so that people will prefer to buy it. If an apple looks round like a tomato, nobody will want to buy it, right? Apple should have its characteristic shape. Then it is more preferred by the customers. So that also here again it is increase in length itself. And yeah, in the brewing industry, to use a substrate for yeast, for the yeast to convert some substrate into alcohol, some substrate is required. One of the substrates that is provided to yeast in the process of fermentation is malted cereals. Okay, so they will allow seeds to germinate, undergo a little bit of germination, and they will heat it a little bit and freeze their growth. That is called malting. So by spraying this, malting also can be sped up. It acts like ole and delays aging. It delays senescence. Okay, so these are some physiological effects that you can see in gibberellins. Next, let's talk about cytokinins. Cytokinins discovery, I'll tell you. They discovered this during tissue culture experiments. There's this one person called F. Skoog and some of his co-workers. They were performing tissue culture experiments. What they noticed was they were working on getting tobacco plants from tobacco stem alone. See, if this is like a tobacco plant, this is the stem. You have the leaves arising from here, let's say. These regions, okay, wherever the leaf arises from, that point on the stem is called the node. The region between the node is called the internode. So they were trying to get an entire plant from just the internodal segments. That is what they do in tissue culture. They take just the leaf part and get the entire plant. They take just the stem, a part of the stem, and get the entire plant. From just the internodal segments, they were trying to obtain whole plants. Okay. So what they do during tissue culture is they will take any part of the plant. It could be leaf, stem, whichever part. Whichever part they take for tissue culture, they call it the explant. And they will grow it in a medium called the nutrient medium, where they only supply whatever nutrients this requires to become this. Right now, before this becomes this explant will become a mass of undifferentiated cells that only know to divide. This is called callus. Okay, first during tissue culture, the explant will become an undifferentiated mass of cells called callus. Later, we have to provide it with certain nutrients that will make it differentiated and become a full plant. Okay. So, while they were performing experiments with this tobacco plant's internodal segments, they noticed that this callus would divide and differentiate only if, along with auxins, one of the following things were added to the medium. It was either extract of vascular tissues like xylem and phloem, or yeast extract, or coconut milk, or DNA. If along with auxins any one of these was not added, the callus would not divide, it would not differentiate. Okay? These chemicals had an effect on cytokinesis, which is cytoplasmic division. During cell division, nuclear division happens, cytoplasmic division happens, right? So after nuclear division, for cytoplasmic division to happen, these were essential, absolutely essential. If any one of these is not supplemented, then cell division would not take place. Okay, because it has an effect on cytokinesis, we call it cytokinins. Now, later, another group of scientists called Miller and his group identified and even crystallized that chemical that was promoting cytokinesis, and they called it kinetin. So cytokinins were discovered as kinetin. So what was kinetin actually? I told you that cytokinins are derived from adenine. Kinetin was basically adenine, its modified form. Okay, that's why, remember, see here, I told you that DNA also could be used. What does DNA have? DNA will have adenine, right? So yeah, that used to be used, and they discovered it from herring sperm DNA. Herring sperm, herring is a type of fish. That fish's sperm, in that sperm whatever DNA is there, from that they identified and isolated the substance. Okay. Eventually, after performing a lot of research and studies in this field, they found out many such chemicals which had cytokinesis promoting activities. Okay, so like in the corn, you had something called zeatin, coconut milk also. So after the discovery of zeatin, there have been many discoveries made which have activities very similar to kinetin. They also promote cytokinesis. Okay, naturally, if they are produced in a plant, where will they be produced? Wherever cytokinesis, cell division is happening. Where does cell division mostly happen? Wherever there is meristematic tissue. Where is meristematic tissue present? At the root apex and the shoot apex, right? Young buds, young fruits, wherever growth is happening, over there, cell division will happen. Wherever cell division happens, their cytokinins will be produced naturally. Now, what are the functions of these? Whatever apical dominance auxins cause, cytokinins will undo that, and they will promote lateral growth. Okay, so if a plant is showing apical dominance, one thing you can do to get lateral shoots is cut off the tip. Otherwise, you can spray it with cytokinins, and that will help you overcome apical dominance. On spraying cytokinins, lateral buds will start branching out. Okay, so it helps in lateral shoot development and overcome apical dominance. Next, adventitious shoot formation. If shoot has to form from any other part of the plant adventitiously, then it can happen with the help of cytokinins. It produces new leaves, and also within the leaves, it will produce new chloroplast. It will promote the production of new chloroplast, and it will make sure that the leaves do not age fast. It delays the process of aging in leaves and promotes nutrient mobilization. If nutrients have to be translocated from one part of the plant to the other, then cytokinins make it easier. So these are some of the functions of cytokinins. Next, let's talk about the discovery of ethylene. This was discovered by Cousins. He confirmed that whenever you keep oranges, like let's say ripe oranges, this will secrete or produce some volatile chemical, gaseous chemical. Now this chemical could make unripe banana. It could ripen unripe banana. Okay, he confirmed the release of this volatile substance from ripened oranges that could hasten the ripening of unripe bananas. Later they identified this as ethylene. This is the gaseous plant growth regulator. Now what are some of the physiological effects of ethylene? It will promote, remember this is one plant growth regulator that has both growth promoting and growth inhibiting properties. Firstly, what does it do? It promotes root growth. We'll start from the root and go on. Okay, root, stem, leaf, flower, fruit, seed, like that. Root. It promotes root growth and root hair formation, which is the growth promoting activity. It also helps in elongation of the petiole or elongation of the internode, specifically in deep water varieties of rice plants. Next, in potato tubers, it will initiate sprouting. Now, all these are growth promoting activities, right? It promotes senescence and abscission of flowers. Now, senescence is aging, abscission is making leaves fall off. This is kind of like a growth inhibiting property. It initiates the production of flowers, specifically in mangoes as well. It breaks the dormancy in buds. It promotes the ripening of fruits, which we saw in case of banana. That is how they were discovered. In pineapple plant, they synchronize fruit set. They make sure that the flowers are produced at once, so that all the fruits can be produced at once, so that the harvest can be done at once. Okay. Remember, for pineapple you have two examples. One is Auxin. It promotes flowering in pineapple. This ethylene synchronizes fruit set in pineapple. Then whenever a fruit is ripening, in certain fruits, the respiratory rate will increase as they ripen and they will enter a phase called respiratory climactic. Okay, the cause for that is ethylene itself. It breaks bud dormancy also, it breaks seed dormancy also. In peanuts, it initiates germination. Okay, breaking dormancy means initiating germination only. Okay, so these are some of the physiological effects of ethylene. Next, if it's gaseous, how is it used on the plant? It is used in the form of a chemical called ethefone. Ethefone is an aqueous solution of ethylene. When this solution is sprayed on plants, it will get absorbed immediately and then it gets transported very slowly inside the plant. This is how they use ethylene on plants, farmers. Okay, so this is basically an aqueous solution. It is readily absorbed and transported within the plant and very slowly released as ethylene. What can ethefone do? In tomatoes and apples, it can increase fruit ripening, which is its primary job, right? That is how it was discovered. In flowers and fruits, it can accelerate abscission. Excuse me. In cucumbers, it promotes female flowers. If there are more female flowers, then you get more fruits, because from the ovary of the female flower itself, fruit will be formed, right? And it shows some influence on dicot seedlings. What it does to dicot seedling, I'm not saying this is economically important, But this is an effect that ethylene shows on dicot seedlings. So whenever there is like a dicot seedling, the seedling will grow horizontally like this. It will grow very thickly and the tip will be curved in the form of a hook like this. These are three effects: horizontal growth, thick, and forming a hook. Okay, so it grows horizontally first, it is thicker, will appear swollen, and thirdly, at the tip there is a hook formation. Okay, now finally, abscisic acid, last phytohormone. Discovery of abscisic acid was not very interesting or anything. Like 3 different people across 3 different parts of the world discovered the same chemical, abscisic acid, and gave it 3 different names. One called it inhibitor B, one called it abscission 2, another one called it dormin because it induces dormancy of seed. And later they identified all 3 to be the same chemical and then they called it abscisic acid. Okay. So this also is called a stress hormone in plants because whenever the plant is under stress, this will come and take care of the plant. All right. So what is the stress for the plant? It does not have to study, it does not have to go to work. Stress for the plant is whenever there is an infection, that is stressful. Whenever there is not sufficient nutrients in the soil, not sufficient water, that is stressful. Whenever the temperature is very high, that is stressful. Whenever it's very dry, that can be stressful. So under such conditions, when the plant is stressful, this plant hormone will do whatever is possible by it to make sure that the plant survives and continues to reproduce and continues to continue the species. Okay. So firstly, it will stimulate stomatal closure. Why will it stimulate stomatal closure? It's a bad thing, right? How will the plant get carbon dioxide if the stomata is closed? If the plant is losing more water than it can absorb, then what is the point of keeping stomata open, right? In such cases only it will close the stomata, not when the conditions are normal. Okay, so whenever conditions are not favorable, whenever the temperature is very hot, the atmosphere is very dry, when the plant is losing a lot of water, then it will close the stomata. Okay, next. It is very, very, very important when it comes to seed development, the maturation of the seed and the control of its dormancy. Abscisic acid plays a very, very important role. It inhibits seed germination, which means it promotes dormancy. Okay. Yeah, why does it promote dormancy? Because if there is very little water available or too dry conditions, whatever water is there in the seed, that also could be lost. The seed could get desiccated and become non-viable. It cannot germinate at all. That can happen. In order to prevent that, it will make it dormant. Okay. Yeah, basically it increases tolerance to stress that I mentioned. Generally, it works against gibberellic acid. Whatever gibberellic acids do, this will do the opposite of it. So we call it the antagonist to gibberellic acid. That is our last point. So with that, we have studied the physiological effects of all the plant growth regulators. Now, these plant growth regulators can work individually or they can work together synergistically. Whenever they are working together, they can complement each other or they can be antagonistic against, like for example, if you see auxins and cytokinins, both of them are promoting cell division. So both of them are used in the process of tissue culture to promote cell division, cell differentiation. If you see auxins and cytokinins with respect to apical dominance, auxins promote apical dominance, cytokinins help overcoming apical dominance. In that case, they are not working together, but they are working against each other for the same function. That is called antagonistic. And then if you consider ethylene and abscisic acid, ethylene promotes seed germination, abscisic acid inhibits seed germination. With respect to seed germination, they both are antagonistic. Okay, they can work, they can have independent functions to perform, they can function together also. If they are influencing the same process, the same phenomenon, then they can both influence it positively, or one can be positive, one can be negative, anything can happen. So I have given like a quick brief overview of the entire chapter with as much detail as possible. I hope this was helpful. So I will see you in the next week with the next chapter. We will be starting the 12th grade syllabus. in the next week. All right. I'll see you in the next class. Thank you very much. All the best.